Okay, you're welcome to this class. This is qualitative analysis in chemistry. In chemistry, we have two types of analysis, quantitative and qualitative analysis. In quantitative analysis, we measure. Example is our acid-base titration, redox titrations. But in qualitative analysis, it is a test to identify cations and anions in salts. In identifying cations and anions, common cations normally students are asked to identify is copper 2 ion, lead 2 ion, aluminium ion, and other ammonium ion. And common anions in salts are chloride ion, tetrodosulfate 6 ion, SO42 minus, SO32 minus, trioxosulfate 4 ion, triosonitrate 5 ion, chloride ion, iodide ion, and many others. In identifying cations and anions in a given salt, you can do preliminary tests. I have samples of salt here. The color of a salt can give you idea of the cation present in a salt. For example, the first sample in my hand is a colorless salt, is a white salt. This salt might likely contain lead 2 ion, aluminium ion, calcium ion, or zinc ion. The second salt in my hand is a blue salt. Most copper salts are blue in color, except copper 2 chloride that is green. So if not, most of the copper 2 salts are blue in color. So this particular salt, when you look at it, as a student, you can assume that the cation here is copper 2 ion, although you've not done the confirmatory test. The third salt in my hand is the green salt. The likely cation you can infer in this salt is ion 2 plus. And another salt I have in my hand is a reddish brown salt. Most ion 3 salts are reddish brown in color. This is what we call preliminary test. Now let's move on to the confirmatory test. Our class today, we are looking at this blue salt. How do I identify the cation and the anion that made up this particular salt? In this particular practical, we have the following apparatus. We have our boiling tube in case we need to boil any solution. We have our test tube. Then we have our test tube rack. We have our reagent bottles. We have our distilled water in our wash bottle. We have our spatula. And then we have our droppers, the dropping pipettes. We have our litmus paper. Sometimes we need to check whether the solution is acidic or alkaline according to the instruction given in the question. So let's start. The first instruction is to dissolve 1 spatula full of this sample. Remember, we don't know the cation, but we presume that copper 2 plus is here. You pick 1 spatula full, add it to your boiling tube or your test tube. So you can use either of the two, depending on the instruction. I'm adding, as you can see, I'm adding a spatula full of the salt into the boiling tube. Add about 10 cm cubed of distilled water. Since it's a qualitative test, we don't measure the water, but you just add a little quantity. The more concentrated the solution is, the easier, the faster is the rate of reaction. You dissolve it. Shake the solution properly to check whether the salt will dissolve in water or not. Anytime you are asked to add water to a sample, your examiner wants you to check whether it will dissolve or not. As you can see, the salt is a soluble salt. Add more water. Remember, the more the quantity of solvent, the higher the solubility of salts. How do you report this? You report this that this is your test, observation, and inference. You draw your table. The test is, you write, test number 1, 1 spatula full of sample A plus 10 cm cubed of water, as you can see in the result table. and record your observation. Sample A dissolved completely in water to give you a blue solution. As you can see, the solution is blue in color. You record it. And what inference? What will you infer from your observations? Sample A is a soluble salt. And since the solution is blue in color, copper 2 plus is likely present in the salts. Now let's go to the next test. The next test is test the resulting solution with litmus paper. You pick your red and your blue litmus paper, slant the solution, put the blue litmus paper and check. As you can see, the blue litmus paper turns red. Record it in your observation. Solution turned, blue litmus paper red. The inference is the solution is acidic. And also check with the red litmus paper. Can you see? The red litmus paper remained red, so it has no effect on the red litmus paper. There is no point reporting this. You report what you've observed. The solution turned blue litmus paper red, and the inference is that the solution is acidic. The next instruction in the question is divide the resulting solution into 3 portions. Remember, I said in qualitative, we don't measure. Take 3 portions out of your solution. Divide it into 3 portions. Okay. We've divided according to the instruction. To the first portion of the solution, the next instruction is add sodium hydroxide in drops and in excess. There are 2 common reagents we use in identifying cations and anions. These reagents are precipitating agents. They precipitate insoluble metallic ions from their aqueous solution. They are sodium hydroxide solution and ammonium hydroxide solution, aqueous ammonia. So whenever we ask you to use them, we want to identify cations and anions. The first instruction, to the first portion of the solution, add sodium hydroxide in drops. Each test must be carried out and the observations recorded. You just need about 2 to 3 drops. As you can see, there is the formation of a blue gelatinous precipitate. Copper 2 ion forms a blue gelatinous precipitate with sodium hydroxide, which is the formation of copper 2 hydroxide, which is insoluble. So you record in your observation blue gelatinous precipitate. You can also abbreviate your precipitate as PPT in small letter. The inference is Cu2 plus copper 2 plus present, as you can see in the table. The confirmatory test, add sodium hydroxide in drops and then in excess. Each step is followed. I'm adding in excess, pour it in excess quantity. You can see that I'm adding excess quantity. The essence is we want to see whether the precipitate will dissolve or not. Shake it properly and check whether the precipitate will dissolve or not. You can see that the precipitate is insoluble. If a precipitate dissolves, it's going to give you a clear solution. But once the solution is cloudy, you can see that the precipitate is settling down at the bottom. So your inference is PPT is insoluble. And our inference is copper 2 plus is confirmed. We'll move to the second portion of the solution. Like I told you, we can still confirm the presence of cations in solution using aqueous ammonia. It is a precipitating agent. It forms insoluble, it precipitates insoluble metallic ions from their aqueous solution. Add aqueous ammonia in drops. You can watch me. I'm using my dropping pipettes to add in drop. 2 to 3 drop is okay. You can also see the formation of a pale blue precipitate. The inference also is that copper 2 plus is present. Now in excess, remember I said when we add reagents in excess, we want to check whether the precipitate will dissolve or not. Okay, shake the solution properly. I'm sure you are noting the observation. Shake it properly. You can see that the precipitate dissolved to give us a deep blue solution. This is because of the formation of a complex compound with aqueous ammonia, tetraamine copper 2 ion. That is what is giving you the deep blue color. It is a confirmatory test for copper 2 plus. So the inference is copper 2 plus is confirmed. So as you can see, we have established it that the cation present in this given salt is copper 2 plus. Let's try to identify the anion present. As you can know, every salt contains a cation and an anion for simple salts, except for double salts that have 2 cations and 1 anion. To the top portion of the solution, you add barium chloride. Like I told you, the method of identifying cations and anions, we use precipitating agents. Add barium chloride solution to the salts. You can notice the formation of a white precipitate. So you can add more to note the white precipitate in the salt. Formation of a white precipitate, the barium chloride has precipitated out barium trioxosulfate 4 and barium tetraoxosulfate 6. So our inference is that SO42 minus or SO32 minus is present in the salt. We are not sure yet of the particular one present. The confirmatory test, we try to check whether the precipitate will dissolve or not. To do that, we add hydrochloric acid in excess quantity to check whether the precipitate will dissolve or not. Whenever you want to check whether a precipitate will dissolve or not, make sure you shake the solution. As you can see, the white precipitate is still there. And this shows that the anion present is not SO32 minus, it is SO42 minus. Alright students, we've established the cation and anion present in this given salt. You've been able to identify that the cation is copper 2 plus and the anion is SO42 minus. So what is this salt? This salt is copper 2 tetraoxosulfate 6 salt. See you next class.